This section of Chapter 4 will explore bacterial growth in laboratory conditions. In most laboratories, including ours, bacteria are grown in nutrient broth test tubes or in nutrient agar plates. This is what we call a closed culture or a closed batch culture. In a closed batch culture, the medium is inoculated with the bacteria and the medium is then incubated. As the population increases in numbers, no new nutrients are added to the medium and waste is not removed, which means that nutrients will be depleted over time and waste accumulates over time. As those conditions continue to deteriorate, the population of bacterial cells will go through some distinct phases. Initially, when the medium is inoculated, the population goes through a lag phase. Lag phase is the phase at which the numbers in the population remain the same. Bacteria have been just inoculated into the medium and they're getting used to the new environment. Synthesis of macromolecules required for multiplication take place. The length of the lag phase depends on the medium and environmental conditions like temperature. In the lag phase, the number of cells do not increase. Slowly, the population leaves the lag phase and cells begin to replicate. The entire population enters the exponential or log phase. In the exponential or log phase, the entire population divides at a constant rate at the generation time. This is the phase of growth at which the entire population is the most sensitive to antibiotics because the entire population is metabolically active. It is also during this phase that primary metabolites and secondary metabolites are produced. During the log phase, cells divide at a constant rate. Generation time can be measured as the entire population doubles in numbers every generation time. This is the phase at which the entire population is the most sensitive to antibiotics since the population is metabolically active. This is also the best phase to gram-stain organisms or to conduct biochemical tests. During the exponential or log phase, primary metabolites and secondary metabolites are produced by the population of cells. Primary metabolites are the end product of metabolism of the cells in the population. Alcohols, vitamins are some of these primary metabolites. On the other hand, secondary metabolites are produced later in the log phase, and they signal a phase in which waste has begun accumulating and nutrients begin to deplete. Therefore, the entire population is in a survival mode as the environment becomes increasingly harsh. This is the phase where antibiotics are often secreted. Following the log or exponential phase, the population enters the stationary phase. The stationary phase is a phase of no net growth of the population, as the numbers of organisms dying and the number of organisms dividing equal each other. In the stationary phase, the supply of energy has been exhausted and waste products are accumulating. Following the stationary phase, the death phase. Death phase is exponential, meaning the number of organisms dying doubles in numbers every set length of time. So the entire population's numbers decline. The phase of prolonged decline follows the death phase. This is a theoretical phase in which mutants arise from the population which will survive the harsh environment. lag phase, initial phase, in which the population has just been inoculated into the new medium, and the organisms are getting ready to divide. Log or exponential phase is the phase of exponential growth in which the bacterial population doubles in numbers every generation time. Stationary phase, it is a phase of no net growth in which the number of cells dividing and the number of cells dying equal each other. Death phase is also an exponential phase in which double the numbers of organisms die every set amount of length of time. Following the death phase, the phase of prolonged decline signals the survival of mutant cells which can live under the harsh environmental conditions. You should now be able to answer the following questions. Describe each of the phases of growth. Which phase of growth is the best at which to do a gram stain on a culture? Distinguish between primary and secondary metabolites and at which phase are each